Good morning, ladies and gentlemen, and welcome to the AVACTA PLC Interim Results Investor Presentation. Throughout this presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted at any time via the Q&A tab situated on the right-hand corner of your screen. Simply type in the text in the box below and press send. If you're accessing this via a mobile device, it will be underneath. Due to the significant number of investors on the call today, the company will not be in a position to answer every question it receives during the meeting itself. However, the company will review all questions submitted today and publish responses where it's appropriate to do so. These will be available via your Investor Meet Company dashboard. I'd also like to remind you that this presentation is being recorded. Before we begin, I'd like to submit the following poll. I'd now like to hand over to Alistair Smith, CEO, Tony Gardner, CFO, and Neil Bell, Chief Development Officer of AVACTA. Good morning.
Morning. Thanks very much indeed, and thanks everyone for making the time to listen to the presentation. I'll push straight on with it so we don't lose time for questions. So for those of you who are new to the company, I suspect many of you will know this, but let me give a quick introduction for those who are new to the company. So we have two divisions, one based in Cambridge, UK, just focused around the therapeutic developments based on the AFIMA platform and the Precision platform. We'll have some time today to talk about the first of those precision prodrugs, AVA6000, that we're pushing into the clinic early next year. And the diagnostics division, which is based in Wetherby in Yorkshire. And of course, we will be talking in some detail around the lateral flow antigen test we are developing with Cytiva and the test developing with Deptrix as well. I'll give you an update on that too. Okay, so brief introductions. Myself, Alistair Smith, for those of you who don't know, Chief Executive. I have a scientific background, but been out of science, as it were, for many years now. And Tony, would you like to say a quick word?
Yeah, good morning. I joined AVACTA back in 2016 as a CFO. I've worked across several different businesses as CFO over the last 20 years, particularly businesses where we've got lots of intellectual property to handle. Thank you.
And Neil.
Hi, thanks. Yes, I've just recently joined AVACTA Life Science as a Chief Development Officer in August of this year, and I'm delighted to be here. So I'm responsible for late-stage preclinical and clinical development for the therapeutics pipeline, including the precision prodrugs and AFIRMA immunotherapies. My drug development career has spanned over 30 years across various leadership roles, working for successful mid to large pharma companies as well as biotech at the global level. And I think it's worth actually highlighting that my drug development experience extends across small molecules, biologics, and more recently cell and gene therapies, where I've taken actually CAR T cells successfully into the clinic. So I've got extensive early and late development experience covering preclinical, phase 1, 2, and 3. And in addition to the technical side of my drug development experience, I've got significant experience of building new organizations from the ground up. So I hope that gives you a useful insight into my expertise. Thanks.
Thanks, Neil. I'd just like to say publicly welcome as well, Neil, to AVACTA, and thank you for the impact, the positive impact you've already had on the company. So really looking forward to Neil's contribution over the coming years. Okay, so let's go through the results and then we'll look at a business update. I'm not going to read through all of the operational and financial highlights because those are sort of historical RNSs that you should all be familiar with. So let me push through to the numbers and let Tony just take you through those.
Alistair. So this is our first reporting period to the end of June, having changed the year end to December back in December 19. So reporting the 6 months to June 2020 compared to June 2019. So revenues have increased across the period, primarily from the custom projects in our diagnostic sector increasing, but also the development projects with our partners such as LG Chem and Day One, where we've got funded income coming through on those projects, which has pushed revenue up in the period. You'll notice there's a line where R&D and amortization costs have increased over the period as we start to build towards taking our first program into clinical trials, and we'd obviously expect those to continue to increase given the cost of that program. The taxation positive numbers there reflect the R&D tax credits which we receive as part of the process we go through on our tax returns every year. And our retained loss for the period was just shy of 7 million compared to 5.7 million in comparative period. The loss per share has reduced slightly given the increase in the average number of shares in issue over the reporting period. Moving on to the cash and balance sheet, there was a net inflow of cash of just over 45 million within the 6 months. Obviously, that was driven by the proceeds from the 2 placings, but also from the purchase of shares by staff from the employee share option schemes that they've exercised. So there was a net 51 million that came into the business in that period. That left us with just over 54 million of cash at the end of June. Now that will be used to fund the expansion of the therapeutics pipeline and also the expansion in the diagnostics side of the business. And that should provide us a cash runway through into 2023. We finished the 6 months with net assets of 71 million, so we're in a strong position as we head forward. Thank you.
Okay, thanks, Tony. So again, I'm not going to read through the post-period highlights. Obviously, we're going to cover a lot of this during the presentation and a forward look as well. So going on to the business update, the first thing I thought I would do is set out some near-term milestones over the next 3 and 6 months. And I'd put those, broadly speaking, into 3 buckets. So the first relates to AVA 6000, which we'll talk about more in a moment. So that is the prodrug form of doxorubicin, the precision form of doxorubicin. And we are aiming for a regulatory filing before the end of the year, with dosing first patients late in Q1 next year. And we should see the initial PK and safety data around the middle of the year. Neil will take us through that in a little bit more detail in a moment. That's a really significant set of milestones for the group, because not only does that move the AVA 6000 into the clinic and transition AVACTA into a clinical stage biotech, but most importantly, I think, should deliver proof of concept for the precision platform in human. And of course, that can be more broadly applied to a wide range of chemotherapies. And again, Neil will talk a little bit about that in a moment. And then the second area, which is obviously of huge importance and very great interest to everyone at the moment, is the rapid antigen test, the saliva-based SARS-CoV-2 spike protein test. We are, and we'll talk through this more, we're aiming for clinical validation in the 4th quarter of this year. I'll talk to you about how we can more precisely nail that down in terms of timing. I'll talk to you about the product strategy in a little bit more detail than we've done in the past around professional use and consumer self-testing. And I'll say a little bit about the additional opportunities that the AFIMAs we've raised against SARS-CoV-2 spike protein have generated. Then the third bucket, as it were, is around the core business. The continued progress of the diagnostic business towards licensing deals for non-COVID applications, which is absolutely key to the progress of that business towards validation of the AFIMA platform in this sector and profitability, notwithstanding obviously the huge COVID-19 opportunity that there is, and of course in therapeutics, where we continue to carry out the business development to deliver further substantial licensing deals for the group. Okay, so let me ask Neil if he would take us through the next few slides around AVA 6000, the first of these precision prodrug chemotherapies, and the taking that into the clinic. Thanks, Neil.
Great. Thanks, Alistair. So I'm going to talk about the AVA 6000 early clinical development, and I think it's important, and Alistair's already indicated this, that AVA 6000 will be AVACTA's first entry into the clinic. It'll be a first-in-man study, and it'll take AVACTA through a really important milestone to become a clinical stage company. So this morning I've got a few slides to cover 4 key areas. The first is, what is a precision platform and how does it work? The second is, what is doxorubicin, also known as AVA6000? The third is the overview of the timeline and design of the AVA6000 clinical study. And finally, the potential for the precision platform to be used across other chemotherapeutic agents. So if we go into slide 15, sorry, that was slide 15, yeah. Okay, so fibroblast activation protein alpha, also known as FAP, is an enzyme known to be highly expressed in most solid tumors compared to normal healthy tissues. So the precision platform is based on using the overexpression of FAP alpha to cleave or to break a link substrate which is attached to the drug and thereby switch the drug into its activated form. And that's why we call this a prodrug. So the graph at the top right hand of the slide highlights the high level of expression of FAP across different tumor types compared to the matching serum. And what this actually means is that the FAP expression is actually localized in the microenvironment of the tumor itself, in the stroma. So precision is a simple peptide substrate which is chemically linked to a drug of choice. And in our case, for AVA6000, it's doxorubicin. The linked drug is actually prevented or limited from entering cells, and it therefore renders the chemotherapy agent inactive once it's infused into the body prior to reaching the tumor microenvironment, where it's activated by high levels of FAP, cleaving the linker to the cytotoxic. And as we know, the substrate is exclusively licensed from Tufts University in the US. So going on to slide 16. So while doxorubicin is an effective anti-tumor agent, it does cause what we call cumulative and dose-dependent cardiotoxicity. So it ranges from hidden changes in the heart structure and function to severe cardiomyopathy and congestive heart failure. And these are 2 serious pathologies, and they may result in even a heart transplant or death. So despite doxorubicin being considered an important standard of care treatment for many cancers, which it is, its use in patients is limited by its cumulative toxicity. And what that means is that physicians are limited by the additive dose of doxorubicin, by the total dose that they can give to patients. Normally, they're limited up to 400 mg per meter squared. So what we have with AVA6000 is we have the potential to address these key cardiac safety concerns for doxorubicin by limiting doxorubicin getting into cardiac cells, to cardiomyocytes as they're called, and actually damaging the heart. So we know that data from mouse models has demonstrated that AVA6000 compared to doxorubicin has significant increase in penetration in the tumor tissue compared to the cardiac tissue. And this means potentially that higher doses of AVA6000 can be tolerated systemically in the body without risking cardiac toxicity. So this is an important market opportunity for AVA6000, and it's significant across a number of tumor types, including soft tissue sarcoma, breast, and ovarian. So in terms of our planned phase 1 clinical study, these are the timelines that we're looking at the moment. We're aiming to file a clinical trial application to the MHRA by the end of 4th quarter 2020. And our team are actively putting all the relevant documentation together to actually compile the CTA now. So our anticipated dosing of first patient in the current planned study is by 1st quarter of 2021, followed by initial safety and pharmacokinetic data from our first dose, first cohort, expected round about mid-2021. So the phase 1 clinical study has been designed in 2 parts. Part A and Part B. Part A is the dose escalation stage, where the main objective of the study is to assess safety and tolerability of AVA6000. And the next objective is to determine the maximum tolerated dose that the patient can tolerate. And that might well be the recommended dose or maybe a dose below. So we anticipate Part A will recruit approximately 15 to 20 patients through approximately 4 dose levels, depending on dose-limiting toxicities that we'll see in the study. Patients will be recruited across a range of cancers, but they'll include soft tissue sarcoma, breast, and ovarian, as we said earlier. Once we've decided on the recommended dose to move into Part B is the dose expansion stage, where the main objective of the study, again, is to assess the safety and tolerability of AVA6000, but in a larger cohort of patients, in possibly 1 tumor or maybe 2 tumor types, depending on the data that we see from Part A. Now, I think it's important to say that, unfortunately, we'll be starting this study in hospitals in the UK, where at a time of COVID-19 pandemic, and where many clinical trials, particularly oncology trials, have been severely affected in the lockdown. We'll monitor the situation and we'll take mitigation actions where necessary to actively move AVA6000 into patient recruitment. And we are looking at the planning of an IND as well in the US. So in terms of a little more detail around the Part A, we actually are going to follow a classical 3+3 design for Part A. 3 patients will be dosed, and if no dose-limiting toxicities are observed, we'll then escalate to the next dose until we reach the maximum tolerated dose. If we do see any dose-limiting toxicities, then we'll need to expand that particular cohort by adding further patients. So we're aiming to dose patients up to 6 cycles of AVA6000 every 3 weeks, which should take normally 18 weeks. So what's the wider precision prodrug opportunity? Well, the wider opportunity and the rationale for precision platform, I think, is compelling. Many chemotherapies have dose-limiting toxicities which restrict their use in patients. Yet despite these toxicities, these chemotherapies retain significant market share and continue to increase their share annually due to better cancer detection rates. As an example, Takeda's Velcade is a protease inhibitor, and its anti-tumor potential has never really been fulfilled in the market due to dose-limiting toxicities, and is only has market approval for multiple myeloma and mantle cell lymphoma. If we look at another example, taxanes such as paclitaxel also has significant sales potential through to 2025, but its market share is constrained due to significant toxicities such as myelosuppression and peripheral neuropathy. So we can see that AV6000 will help us to demonstrate the value and the potential of the precision platform. We're at a very exciting junction with precision, and we need to continue to drive towards the milestones that have been laid out in the development plan. So I'll stop there and hand back to Alistair.
That's great. Thank you, Neil. Okay, so pushing on, because I'm very sort of aware of time and wanting to make sure that we have time for plenty of questions. So the first thing I wanted to do was talk to you about the performance of the Affimer reagents that AVACTA's developed against the SARS-CoV-2 virus spike protein. So these are the Affimers that are in the lateral flow antigen test and in the Adeptrix test, and potentially can be licensed to other third parties for their tests as well. So the point that I want to get across is that we now have a fully validated, what's called an ELISA test, which is a laboratory test. We will be launching this as a research laboratory test as soon as possible. But the point I wanted to get across is the performance of the Affimers is really excellent. This is, as far as I can find, certainly on the internet, the best performing ELISA test that we can see. both in terms of sensitivity in simple systems like buffer, which is effectively just water, as well as in saliva. We've not been able to see any saliva performance data for other ELISA tests on the internet, but also in terms of specificity. So very briefly, because I appreciate that not everyone has a technical background, but let me try and put this in context. The sensitivity we can talk about in terms of the lower limit of quantitation. So that's a term used to describe the lowest amount of spike protein that can be properly quantitated in a sample. And that is down at a few picograms per mil. So to put that in context for you, that is very good for any ELISA. It's certainly much better than other SARS-CoV-2 spike ELISAs on the market. And I've given one there just for reference. You can go to the website, you can see the performance of that test and make the comparison yourself. But also to put that in context, that level of detection, that level of quantitation is right down at the lowest levels of viral infection. And the work we did with Liverpool School of Tropical Medicine to confirm that shows that this ELISA test will detect down to just a few thousand copies of the virus per mil of saliva. And you see there the saliva performance in the graph and the table as well. So from a sensitivity perspective, this gives us huge confidence in terms of the performance of the lateral flow test and the Adeptrix test and the others. The AFAMA reagents work really well. Specificity is absolutely key, as we'll see in a moment. And you see there, without going into a lot of the detail, no cross-reactivity against common coronaviruses and less common ones like SARS-CoV-1, which is just not around too much anymore. So no cross-reactivity, very good specificity as well. So that's the performance of the basic AFAMA reagents. Let me touch on now the BAMS assay. So this is a mass spectrometer assay, and I've done a very simplified cartoon of how this works. So the sample introduced into the sample are beads coated with the AFAMA. So those will capture the spike protein and virus from the sample, and then you can use a magnet, obviously, to hold those whilst you wash off all the other stuff that will be in that sample. And then those beads can be introduced into the mass spectrometer in a very simple way. And one of the beauties of the way in which Adeptrix has developed this test is that there's a very simple yes/no answer that can be generated from what is quite a complex mass charge spectrum. You'll see in a second on the next slide. So that's fundamentally how the test works. And in this way, each machine can do 1,000 samples or upwards of 1,000 samples per day. And of course, the installed base of these machines is quite considerable across the UK and elsewhere in clinical microbiology labs in hospitals. So that's the opportunity for us to deliver. And we haven't been able to show much data so far, primarily because of commercial confidentiality with Adeptrix, but and I obviously don't plan to talk through this mass spectrum. But for those of you who've never seen these things before, that gives you an idea of, let's call it, the fingerprint that this test delivers. And there's 2 points to make. So first of all, this is the mass spectrum from one bead, a single bead. So in terms of sensitivity of this test, it's excellent. And then in terms of specificity, you are combining the AFAMA reagent specificity to the SARS-CoV-2 spike protein with this fingerprint that is literally a fingerprint of the protein. So you get exquisite sensitivity and just an extra bit of detail. This spectrum gives you information about things like mutations of the virus as well, potentially. So not only a diagnostic, but a really important tool. So what we are literally all of us trying to get to is the clinical validation data. And without going into a lot of detail, it has been a frustrating process getting through the UK Health Systems Condor system to get that clinical data. We are making progress, but it's been slow. However, what is a real positive is the level of commercial interest we've now got for this test, and it's clearly critical that we have a significant partner who is most likely a mass spectrometer manufacturer who can take these BAMS assays to market, both for research use and as an IVD, and get those into the install base at scale to generate the commercial opportunity for us. And I'm very pleased to say we've made solid progress in securing that commercial partnership, which I hope over the next weeks and months we'll be able to talk about publicly. Okay, so that's a quick update on the BAMS assay. We are getting on with the clinical evaluation now, but it's still slow. So as soon as I have some clinical data to talk about, I will do. But we're obviously relying on third parties for that. So then getting on to the lateral flow antigen test. I've shown this slide before, so I won't dwell on it for too long. But it's for those of you who haven't seen this slide, it's really understanding the difference between an antigen test, which tells you whether you have the infection currently, or an antibody test, which really tells you, and we'll see on the next slide the detail, but really tells you whether you had the infection in the past or not. Two different functions, two different uses, but what we're really focused on, and what certainly the UK and worldwide governments are focused on, is identifying infectious individuals. And this next slide really gives you a bit more insight into that. So this is a figure taken from Nature, and what it shows you is the sort of relative utilities of PCR antigen testing and antibody testing, and you can see that the PCR and antigen testing have the best likelihood of getting an accurate result early on in the infection, when there's a large amount of material to detect, and it takes probably 7 to 10 days post-symptom onset for the antibodies to be generated. So the benefit of antigen testing compared with PCR is that you don't get that long residual positive result long after the patient has recovered. And antigen testing is most effective in that period of a couple of days, 2 to 3 days before symptom onset, and up to a sort of week or so after symptom onset, when the patient has the highest viral load and is most infectious. I'm going to touch on that again on this slide because it is so important for the intended use of these tests. So again, I've put some references here, which if you have time, you can go and have a look and read a little more around the detail. But the presence of the virus, the infectious material, is highest in the first couple of days, sort of 5 days or so after you get infected, the first couple of days before symptoms are seen, and then the next sort of 7 to 10 days after symptoms appear. So there's a window there, which is when the antigen tests work particularly well. And of course, it's that period when individuals are infectious. So there's a very short period when you get infected but are not necessarily infectious, because there's not enough viral load. There's not a virus in your upper respiratory tract to be shedding it everywhere and infecting other people. And so the purpose of these tests is to identify those people who are infectious, not necessarily all people who are infected. And just interesting point to note that that pre-symptomatic period of 2 or 3 days before symptoms appear is thought to account for around 50% of infections. So that is why this is so important, to be able to identify infectious people who are pre-symptomatic. And sometimes those people are referred to as asymptomatic incorrectly, actually. The number of people who are truly asymptomatic throughout the infection is it's actually difficult to determine, because obviously they're asymptomatic and there's a lot less data around that. But certainly pre-symptomatic transition is a major issue that antigen tests can address. Okay, so then again, just making the final point on, and I've been asked a lot, I mean, a huge amount about sensitivity, and what are you aiming for? And I wanted just to sort of dig into the detail just a little bit for 2 minutes. So what you see with the graph here is, it's just a made-up distribution of patients with different degrees of infectivity, different degrees of viral load. So on the left, those people who have very high viral load, and that may go from just a few virus particles per mil, up to maybe 10 to the 9, a billion virus particles per mil of saliva. So there's a very broad range, and you are infectious probably from around 1,000 or a few thousand viral particles per mil and upwards. So that's that group on the left there that I've shown. The red line shows the difference between not being infectious and being infectious, and everyone to the left of that black line is infectious. And it's absolutely essential to be able to identify those people quickly. So the purpose is frequent testing with a very specific test. You need a very specific test because otherwise you're going to create a large number of false positives, and that will overload the already stretched centralized testing. But this is exactly what the governments around the world are now trying to achieve: frequent testing on a weekly, or even more often, a weekly basis. And the sensitivity within that group has to be very good. You need to be able to identify all of those infectious people. Unfortunately, they're the ones with the highest viral load, so that is more straightforward. If you were then to look at the sensitivity across the entire group, clearly it would be lower. But that's the subtlety and the way in which different companies present these results. And obviously, we will be very clear how we do the clinical validation and exactly what we're measuring and how we express that in terms of sensitivity. But that's the question of infectious versus infected individuals. And the use cases, therefore, look somewhat like this. So the product strategy is to CE mark for professional use first, because that is quicker and more straightforward. It's a self-certification process, as many of you know. The process to achieve consumer self-testing is a little bit more involved and involves what's called a notified body that has to audit your technical file. So that takes a little bit longer. The professional use case, I mean, just to take hospitals from it, immediately that suggests hospitals and GP surgeries. Actually, I think the hospital use of lateral flow tests is not likely to be that high because the PCR is on site. It's relatively rapidly accessed. It's far more likely to be professional use around workplace testing. And that involves having a trained technician or healthcare worker on site, which many companies are planning to do, to be able to do this sort of testing in the workplace with a professional use only test. Testing stations, GP surgeries potentially as well, triaging individuals to reduce the burden on the current, the current testing capacity, centralized testing capacity. And then the use case for consumer self-testing is obvious, of course. It's individuals at home as well as individuals in the workplace, but predominantly it will be testing at home. And then we've talked quite a little bit about the product profile. Saliva, because of the ease of use compared to NP swabs. Visual readout is very important, and we are confident of being able to deliver a visual readout test, which means it doesn't need a specialist reader, which of course, if it did, would limit the deployment of the test. So visual readout is very important. And another area that we've been doing a lot of work on is with our partners is around a smartphone app to read the result, which provides objectivity, which is very important because, of course, this test will limit or prevent certain activities. So objectivity is very important, plus the data connectivity into the healthcare system, data systems, and ultimately provide a record of a positive or negative result. So all of this is going on at the moment, somewhat behind the scenes, but we can update on that as we progress. Okay, so let's just talk through what we've learned. We AVACTA have learned a huge amount about the market and the demand and the use cases that we've talked about. And what I can tell you is that, within the UK alone, in order to have effective disease control, i.e. get this R value well below 1, you need frequent testing with these types of tests. You need to be able to catch the infectious people, ideally as soon as they're infectious. So let's say weekly testing, and ideally it would be more often, but let's say weekly testing. So that, even if you tested half the population weekly, that implies 120 million tests per month, and that's UK only. So that does not obviously include any US opportunity or elsewhere. So clearly the commercial opportunity for AVACTA is determined right now by supply capacity and not by demand. There is lots of room in the market. And again, people ask me very often about other tests that are coming through. There will be other tests. There is more than enough room for many tests, many antigen tests across not just the UK, but the rest of the world. So that competition, if you like, doesn't bother me in the least. The demand is really significant, as we're now very clear about. So the supply capacity is what we need to worry about. And we have been for the last 6 weeks or so, really working very intensively on this. So there are several factors: raw materials, so the nitrocellulose strip made by Cytiva, and the AFMA reagents. We'll talk about that in a moment. The lateral flow strip manufacture and assembly into cassettes. The plasticware, so the saliva collection plastic, and the what's called a transfer pipette to move the sample onto the lateral flow test, and then assembling the full kit. So that's putting all of these elements in a box with the instructions for use and so on, and shipping those. So I'll talk about that in a moment. The other area in which we have been working to add capacity and increase the commercial opportunity for AVACTA is looking at OEM deals. So this is providing the AFMA reagents to third parties who may develop other types of testing modality. And obviously that doesn't impinge at all upon our own capacity, but is a good way to expand that opportunity. And we have several ongoing conversations with potential OEM partners to add to that commercial opportunity for AVACTA. Okay, so let's look at those individual elements that determine capacity. So first of all, we are putting in place, and in fact we will have finished putting in place by November, the increased production facilities within AVACTA. So we will be able to manufacture at reasonable scale ourselves in-house and have control over that. But we're also putting in place very large scale contract manufacturing partners. And we will, I'm pretty sure it'll be a UK-based manufacturing partner that will allow us to scale that up to very large volumes, which if we get to that 120 million per month or even higher numbers, by accessing other markets around the world, that will require some third-party contract manufacturing. So the AFMA supply, we do not believe is going to be a bottleneck, a limiting factor, and nor will the nitrocellulose strip. So we've partnered with BBI Solutions and Abingdon Health to date, as you know, and they can manufacture several millions of lateral flow test strips per month. Excuse me. So clearly there is a significant upscaling that needs to be done within the UK to be able to meet the demand, and that I'm sure we certainly are supporting our partners to do that upscaling. But I think what it speaks to is the requirement to put in some capacity overseas as well to be able to meet that demand. I mean, we will put through all the manufacturing we possibly can in the UK. But we are very actively in discussion with manufacturers overseas, mainly Asia, Southeast Asia, to be able to put in place increased capacity. And that, of course, just means doing the job properly and making sure that we do our due diligence properly and getting quality product manufactured. The kit assembly, whilst you might sort of think that's straightforward, it is a reasonable challenge at these sorts of volumes. If you do a quick calculation of how many kits per second 120 million a month represents, it's a significant number. So we've already identified UK partners, and again we'll be putting in place further resource overseas to be able to not only meet the demand, but hopefully look at other markets around the world in addition to the UK. So we now have detailed discussions ongoing on with government and regulatory authorities, healthcare providers, and so on. But those are strictly confidential at this stage, and I can't talk in any more detail about those. Okay, so as I'm sure you all know, we are in the process of technology transfer. Cytiva and BBI, so our partners, are working together to carry out that technology transfer. And what that involves is the development of the scaled up process to produce the test strips that perform adequately for the what we know is the clinical performance targets. And just to be clear, that is not a simple tick box exercise. That is a process, a development process, which takes some time. And as soon as we have good visibility on that pilot batch of around 10,000 units, which we intend to use for the clinical validation, as soon as we have visibility on that, I will be able to give you all a much clearer, a much more precise timing of the clinical validation, CE marking, and product launch. I think it's important to stress that this process of manufacturing scale up, the tech transfer, it is absolutely essential that we do that properly. Rushing that simply pushes risk down the line to the clinical validation. I'm not prepared to do that. So we are working with really excellent partners to get that process nailed so that we have a scaled up process that is robust and reliable. And that takes out a huge amount of technical risk from the process. So then the clinical validation and lay user studies, which is the study required for the additional study for the consumer self-test CE marking, those are being designed with Liverpool School of Tropical Medicine. That's a relatively straightforward process. We get patient sample access through LSTM and also through the Condor system, and we will complete the verification and validation, and that will allow us to self-certify for professional use, as we talked about. We'll run the lay user study in parallel as much as we possibly can with that process, and then go into the audit by the notified body of the technical file. And we're looking at how we can streamline this process, but keep it robust so that we end up with a product that is competitive globally at identifying infectious people in the society. So as I said, just to summarize that slide, the manufacturing scale-up process is a development phase, and we are going to do it properly, and we are going to take risk out at this stage rather than pushing it down the line. And when we have completed that and got to pilot batch, which won't be too long, then we will obviously be able to give a much more precise picture of the path through to product launch, which is what we all want to see. Okay, so just to summarize and give ourselves some time for questions, and I'll hit the main bullet points here rather than go through them all. So on the therapeutic side of the business, the effect of COVID actually on both sides of the business has been really on the laboratory operations, because of course we've had to maintain social distancing within the laboratories. And that has meant we've effectively been working at half staff to get that sort of half staffing levels to get that social distancing. We are putting more space in place both in Cambridge and in Wetherby, because we see this issue lasting for a considerable length of time from now. So we've actually taken on more space so that we can get everyone back into the laboratories, and we're fitting those out now to be able to get back up to full capacity. As I expect this issue of social distancing to remain for some considerable amount of time through next year and potentially even into 22. So the progress on the therapeutic side has been very good. We focused on partner programs to ensure that those kept moving forward. So I would like to say, and it's a good opportunity to thank the teams publicly for the really positive attitude and commitment that they've shown, despite the challenges of traveling and the risks that has presented. The attitude and the commitment of both teams in Cambridge and Wetherby has been outstanding to keep progress, not only keep progress going, but to grow the company and to create these opportunities under these sorts of pressures. And on the diagnostic side, good progress in getting this COVID-19 lateral flow test to market. We've talked about the next key milestone is the pilot batch, and then I'll really be able to set out the launch date with more certainty. But also remembering that there are multiple additional COVID testing opportunities, not least through the mass spec test with Adeptrix, but also other potential partnering opportunities for those SARS-CoV-2 spike AFIMers. And then finally, obviously, during the summer, we raised a significant amount of capital, and that puts us in a very strong balance sheet position to be able to deliver on the opportunities we've talked about, drive multiple programs into the clinic, and add huge value there, and support the core businesses. So I'll leave it there and give ourselves some opportunity for some questions out of what's been a very, very large number of questions.
That's brilliant. Thank you very much, Alistair, Tony and Neil. Ladies and gentlemen, please continue to submit your questions using the Q&A tab situated on the right-hand corner of your screen. But for investors on the call, particularly for those that came in perhaps registering on the platform slightly after the event had started, that a recording of this presentation, along with a copy of the slides and the published Q&A, will be available via your Investor Meet company dashboard. I'd also like to remind you that your feedback is important to the company. Immediately after the presentation has ended, you'll be redirected for the opportunity to provide feedback in order that the company can better understand your views and expectations. Alistair, thank you very much for dropping on the chat. I guess a selection of the questions from investors that have come in today, and obviously a selection of those that were pre-submitted. And I think just given the number of questions that have come in on the platform, along with the number of investors on the call today, that for any question that doesn't get answered, the company will be reviewing all the questions submitted today, and we will, as I said, publish responses where that is appropriate to do so. So the first question, Alistair, I think you've selected is, could you please give an explanation of Cytiva's role in the manufacture of the rapid antigen test? It was initially indicated that they would manufacture, but this hasn't been mentioned in subsequent news regarding BBI/Abingdon. If so, will they manufacture for the US market only?
Yes, thank you. So that's a good question, allows me to sort of clarify. So Cytiva are a materials company, so they manufacture the materials to make the strip. And so we probably, could have been clearer on that. So they supply the nitrocellulose strip, not the finished IVD. And so the manufacturers who make the finished IVD are the likes of BBI Solutions and Abingdon Health, who we've already partnered with. There's a couple more potential partners in the UK, and as I said, many more around the world that we can access as well. So it's really a process of raw materials from Cytiva and AVACTA. So we manufacture those raw materials. We have partners then who do the strip manufacturing, and then partners who do what's called the kitting, putting the kits together, and then obviously the logistics of shipping those out.
Thank you very much. Follow-on question, what is the maximum number of LFT antigen tests that can currently be produced per month? And what will be the maximum capacity in 6 months? Can you give a ballpark figure for the potential worldwide manufacturing capacity for AVACTA's rapid anti-chest test?
Yes, so I touched on this in the presentation, but in the UK, so we are partnered with BBI and Abingdon to access their available capacity, because of course they are doing other things, and we will look to partner with others in the UK as well to get as much of the available capacity in the UK as possible. I suspect, without giving away anything that's commercially sensitive, I suspect that is 5 to 10 million tests per month as we sit here today. So that can be scaled. And that depends on investment by partners and others to support that expansion, which clearly is in everybody's interests. And it's difficult to put a precise number on that, but I do believe that could be many tens of millions of tests per month. We've talked about requiring in the UK alone, even just for weekly testing of half the population, 120 million tests per month. So that I think does still indicate some capacity overseas. The total capacity overseas is is difficult to sort of know with certainty. We've done a lot of work in Asia Pacific to understand what the opportunities are there, and that there is, without doubt in my mind, the capacity to meet those hundreds of millions of tests per month. Although, as I said, doing the due diligence on any third-party overseas manufacturer has to be done properly. Otherwise, it's a whole world of pain that you have to deal with later.
About what you see as the potential for the two therapeutic platforms and their combination.
Sorry, I didn't catch that, but I think, can you say it again?
Yes, of course. Can you talk about what you see as the potential for the two therapeutic platforms and their combination?
Yeah, it's a great question. So, these two platforms, Precision and Afirma, actually are really interesting in their own right, but in combination are very interesting as well. So the Precision, as I've already said, the wider opportunity and the rationale for the Precision platform, as I indicated, is actually very compelling. Conventional chemotherapy is designed to eliminate that fast-growing tumor cell, but obviously it's also harmful to normal tissues as well. So the Precision platform will actually help create a much more targeted therapy. Then you look at the Afirma platform, and this actually brings really interesting alternative to antibodies, because it's derived from a small human protein. It's approximately 10 times smaller than antibodies. So that offers potentially significant advantages in terms of tissue penetration into solid tumors. And there's a huge amount of work going on at the moment to actually develop drugs that will get into the tumor microenvironment and be more specific. In addition, the Afirma proteins can be actually genetically fused together to form what we call multispecific. And at the moment, there's a huge amount of interest and development activity in actually developing bispecific monoclonal antibodies. And so again, the Afirma approach, with its small size, it can actually, we can design approaches by checkpoint inhibitors. So these are pathways that are set up large to actually, for the tumor to defend itself, because the tumors are very clever when they actually are growing in the body, and they start to actually work out ways to defend themselves. So when you combine these two platforms, combining Afirma's with the chemotherapy agents and using a linker that is designed only to release that chemotherapy agent into the tumor microenvironment, then this actually enables an extremely potent chemotherapy to be combined with an Afirma immune checkpoint therapy. So I think, there's a lot of work going on at the moment with antibody drug conjugates. It's a very exciting area, and I think these two platforms being combined actually helps us to get into that area very seriously.
Thank you very much. Question here, are you looking to license out the rapid antigen tests so that companies worldwide can mass produce it under their own names?
Yes, so we are both looking to license the reagents themselves against the SARS-CoV-2 spike protein, and that allows other diagnostic companies to develop a range of different diagnostic tests on different platforms. But also, yes, specifically, there is the opportunity to license the Afirma to allow other people to develop their own lateral flow antigen tests. And that the reason why that is commercially sensible and interesting is because of the way in which manufacturing capacity limits our own ability to exploit the commercial opportunity. So that gives us access to third-party manufacturing scale. And these companies are, of course, going to develop tests regardless of whether there's an antibody in there or an Afirma. And I want an Afirma to be in those tests, so we deliver as much potential value to shareholders as possible. So yeah, the answer is yes. We are more than happy to license for OEM manufacturing.
That's brilliant. Thank you. Next one, is it intended to make an application for approval and use of the rapid antigen lateral flow test in the US, e.g. FDA, EUA, or applications in the Far East?
Yes, yeah. So we are. We've mapped out that sort of pathway to to go through the FDA process. The one thing I would just sort of add to that is that just pragmatically, you can see the scale of the opportunity in the UK and how we need to work on manufacturing capacity initially before we can really exploit the opportunity elsewhere. So we have that procedure mapped out. It's actually a relatively straightforward pathway, and we can go through that quite quickly once we have a CE mark product in Europe. So yes, we are looking at the US opportunity as well.
That's brilliant, thank you. Next one, there has been perceived delay by the market in getting new products into production. In terms of gaining approvals and credentials for product or products being developed, is this being sought directly by AVACTA or its partners, Civita or Adeptrix?
Yeah, so the lateral flow device is AVACTA, so we are the legal manufacturer of that, and we'll hold the CE mark. So it's AVACTA that will take that through the regulatory process. Then in terms of the bands assay with Adeptrix, we touched on that a little bit in the presentation. So the way our relationship with Adeptrix works is that they would be the legal manufacturer of that. But as I said in the presentation, we're both aligned on the fact that we need to partner this with a large, ideally mass spectrometer manufacturer to develop these kits. And I think I said as we went through the presentation, we've made really good progress in that regard.
Thank you very much. There's a question here around listing. The company's revolutionary development of AFAMA antibodies deserves to be exposed to a wider investor community than in the UK. Is there any intention to list on NASDAQ at any stage?
Yeah, so I agree. I mean, there is significant interest in the US around actually both the AFAMA platform and the precision chemotherapy platform. And so the US capital markets are certainly a medium-term aspiration for the company, but no immediate plans. But we have over the last 18 months or so, and we'll continue to do a lot of work in investor relations in the US, with a potential view to that in the medium term.
Thank you very much. Your SARS-CoV-2 diagnostic methods are based on detecting the S1 glycoprotein. Does the N-glycosylation effect for the binding of the AFAMers, what impact do they have on the migration of S1 through LTF? Okay,
so that's a detailed question from someone who's got some technical background, which is great. So the question is asking is that the SARS-CoV-2 spike protein, that protein is modified in nature normally by adding sugars, so it's called glycosylation. And the question is, does that affect whether the AFAMers bind to it or not? And how does that affect flow through the lateral flow device? So it's an important question, actually, a very detailed technical one, but I'm pleased to say that the binding of the AFAMer reagents is not glycosylation dependent. So the AFAMer reagents work with a range of different glycosylation states, and we can see that from intact mass spectral profiles that we're capturing AFAMers with a range of different glycosylation modalities. So that's not an issue. Whether the different glycosylation states flow in a similar way through the lateral flow device, don't know at this point. We'll be able to study that if we have the opportunity, but we're really focused on really getting the product to market rather than digging into the more interesting, perhaps scientific questions there. But one point I would make, and no one's raised it, but it is important. You may or may not know that there is a mutant of the SARS-CoV-2 virus, which is called D614G. So this is a mutant of the spike protein, and it's about 70% of cases in the US and EU now. It's a more infectious form of the virus, and there's a bit of evidence actually that antibodies generated against the normal virus are not effective against that mutant. So the immediate question is, do your Affimer reagents bind the mutant? Because if they don't, you've got a problem. And the answer is yes, they do. We demonstrated that. So the Affimer reagents bind both forms of the virus spike protein.
Thank you very much. Are AVACTA still talking to pharmaceutical companies with regards to the ACE2 blocking Affirmers being used as a therapy? Yes,
well, thanks for that question. Yes, so we licensed some of those, or the around those binders to Daewoong Pharmaceuticals, our partner in South Korea, with a view to incorporating those into stem cell therapies that would use the stem cell aspect to treat the tissue damage caused by the immune response to this infection, but at the same time would produce neutralizing Affimer therapies to limit the spread of further infection. So we continue to have some discussions with other pharma companies, but just to sort of put things in context, I don't want to sort of over-egg the pudding. You know, to take a neutralizing therapy through into approval would require, on a relatively useful timescale, would require a hugely accelerated clinical development program. And generally speaking, because Affimers have not yet been into human, you know, that represents a significant additional risk. So, you know, I don't want to over-egg the pudding and suggest there's massive opportunities for, you know, for a major pharmaceutical partnership here, but there's certainly interest because of the speed in which we develop those binders, and therefore prove the performance of the Affimer platform very well.
Thank you very much. Alistair, I am mindful of time, obviously, with through the hour, and I know that investor feedback is particularly important to the company. I propose just to ask you this final question and perhaps ask you to wrap up. And as I said before, to remind investors that every single question received by the company will be reviewed. And as I said, appropriate responses will be given where that's appropriate. So the final question, I guess, given the cash injection achieved through the fundraise in June, plus the expected significant revenues from COVID test sales, will AVACTA look to change their strategy with the cancer therapies and take the likes of AVA6000 pro-doxorubicin beyond phase 1 trials prior to seeking partners in order to extract greater monetary value for shareholders?
Yeah, so I think that is a very good question, and certainly you know, dependent on, you know, what could be very significant revenue from the COVID testing opportunity. That does allow us to potentially look at pushing AVA6000 further through clinical development, and Neil is building the team to be able to do that. And I'm totally aligned with the person who asked the question that we, you know, potentially could take AVA6000 further into clinical development and add significant value for shareholders around that particular asset. The other aspect of use of proceeds, as it were, and use of profit from future revenues would be around expanding the Affimer and precision pipeline. And I think Neil very sort of clearly set out the, you know, the potential not only of those two platforms on their own, but also the combination of those in a very novel drug conjugate form. So, you know, we would certainly look to invest, you know, any future sales from COVID-19 testing products, as well as the money we raised during the summer, in expanding that pipeline. I hope to be able to take some time, maybe later this year, maybe early next year, to really talk in detail and communicate the expansion of both the Affimer and precision platforms, the biological and clinical rationale around the choices that we've made, because that was a very clear use of proceeds that we set out in June.
That's brilliant. Thank you very much indeed. Perhaps, Alistair, just finally, I could ask you just to wrap up, and then obviously I'll give instructions of how investors can provide you feedback, if that's all right with you.
Yes, of course. Yeah, no, I'm very grateful for everyone spending the time again. Thank you, Neil. Thank you, Tony, for helping with the presentation. I I think we're all totally aligned in what we're expecting to see over the next 3 months or so, which is the CTA filing around 6,000 and clear progress into the clinic with that, the delivery of a lateral flow antigen test as the highest priority on the diagnostic side of the business, and we're doing everything we possibly can to support our partners to make that happen as quickly as possible. As soon as we have clear path to CE approval and launch, I will make that very clear to the market. Right now, we are focused on that manufacturing scale up to get to a robust product for clinical validation.
That's brilliant. Thank you, Alistair, Tony, Neil. Thank you very much for updating individual investors today, particularly given that this is the day of your results as well, and enabling them to absolutely come to a more informed decision. Could I ask investors please not to close today's session, as you'll now be automatically redirected for the opportunity to provide your feedback. If you access this meeting via our website, the feedback page will appear. If you access this link sent to you by email, you'll be asked to log in to submit your feedback. It will take a couple of moments, but we'd very much encourage you to do so. On behalf of the management team at AVACTA PLC, we'd like to thank you for attending today's presentation. That now concludes today's session. Good morning to everyone.